Okay, hello. Thank you for joining us today. I am Tracy Smith, the Collaboration Education and Outreach Coordinator for the Center for Integrated Cellular Analysis here in New York City. And in this seminar series, we highlight new papers and methods from our center that we believe to be of interest to the broader community. Today, we focus on a new high throughput, single-cell multi-omics platform for linking genotype to chromatin accessibility at single-cell resolution, called, and this is a really fun name, Gotch. It's compatible with a 10x Genomics scATAC-seq kit and is a powerful tool for understanding the epigenetic effects of somatic mutations in human cells, especially when mutant and wild-type cells are mixed together in a single sample. We'll have time at the end of the talk for questions, and we encourage you to either raise your hand and I'll ask you to unmute yourself, or you can type your question into the chat and I'll read it aloud to the speaker. Our guest today is Dr. Franco Izzo, currently an instructor of molecular biology and medicine at Weill Cornell and the New York Genome Center in the Landau Lab. His research focuses on clonal hematopoiesis and myeloproliferative neoplasms, where he aims to link genotypes to phenotypes within human samples by applying single-cell multi-omics. He is supported by the American Society of Hematology Fellow to Faculty Scholar Award. And now, Franco, I'll hand it over to you.
Thank you so much for such a great introduction. So today I'll be talking about our latest work. It's a preprint, which is entitled Integrated Single-Cell Genotyping and Chromatin Accessibility Charts JAK2 V617F Human Hematopoietic Differentiation. It is at this point well understood that cellular differentiation is governed by epigenetic changes. And in particular, we can use chromatin accessibility profiles for example, in this picture of sorted hematopoietic progenitor cells to show cell type specific changes both at proximal and distal regions, which can be in turn used to infer the various stages of differentiations during hematopoiesis, for example. Now, in addition, it has been shown that the chromatin accessibility profiles, these changes actually precede the transcriptional changes that lead to differentiation and specification of cellular fates. And therefore, cell identities and differentiation stages can be readily identified through chromatin accessibility profiles. Now, however, these defining epigenetic features can be corrupted through the accumulation of somatic mutations. And these mutations usually result in what we call shifted differentiation topologies, and thus give rise to an aberrant hematopoiesis. So one of the main challenges when trying to study the impact of these somatic mutations in human patients and in human samples in particular is to identify these often rare mutant cells and distinguish them from the wild-type counterparts at enough resolution to be able to draw significant conclusions. So to tackle this particular challenge, our lab, is focusing on developing different technologies. But usually when we apply the standard single-cell sequencing technologies, which are excellent to resolve cellular heterogeneity, usually the similarities between wild-type and mutant cells can actually obscure the identification of mutant-specific phenotypes through these single-cell approaches. So the critical question that we ask and we try to address when developing these technologies is, how can we capture single-cell genotypes and link them directly to cellular phenotypes? And we're particularly interested in technologies that are, amenable to be applied in human samples in particular. So our lab and others aim to develop these kind of technologies by achieving targeted genotyping in conjunction with single-cell information at single-cell resolution. So one of such methods is genotyping of transcriptomes, or GOT, which enables simultaneous genotyping with high-throughput single-cell RNA-seq. This was published by our group in 2019, and is we think is a very useful method for genotyping, which, however, we recognize has some limitations. So basically, genotyping of transcriptomes, as the name indicates, is based on RNA molecules. So we basically capture mRNA transcripts, and we try to identify the mutation within those transcripts. So by relying upon mRNA, this results in some limiting dependencies, which the most critical ones are actually the gene expression, because of course the transcript has to be present within the cell to be able to be captured and genotyped. And in the case of using 10x technologies, the location of the mutation relative to the end of the transcript is very relevant, because you need to be able to capture the mutation site during the RT step. And this example can be seen in our GOT paper, that when we applied genotyping of transcriptomes to CALR, we obtained a genotyping of 85%, which is shown on the top left here. And CALR is relatively highly expressed, and the mutation itself is relatively close to the 3' end of the gene. So when we try to apply genotyping of transcriptomes to, for example, a different mutation in JAK2, the genotyping efficiency drastically drops. And this is mostly due to the fact that the JAK2 gene itself is lowly expressed, so it's very susceptible to dropout, and it's more challenging to actually capture the mutation from the transcript. And it has the additional limitation that the mutation itself is far from 3' end, so it reduces the chances of the RT step actually capturing the mutation site. So then we observed that the genotyping efficiency dropped from 85 to 7% when using this method. So to try to overcome these limitations, we developed genotyping of targeted loci together with Chromatin Accessibility, which we call Gotch for short. And this method is also based on the 10x genomic platform, but in the single-cell ATAC platform in particular. So the first challenge was to capture genotypes within in-droplet cell barcoding PCR. And that uses the TN5 extra adapter sequencing for priming. So to achieve this, we added 2 locus-specific Gotch primers, which are shown there in red in step 3, that flank the locus of interest, with 1 primer handle containing the Nextera read adapter sequence, which you require for capture in the bead, and it's the same way that you capture the segmented chromatin fragments, for example. But further, we need to enrich this genotyping molecules out of a multitude of ATAC fragments. So what we did was to add a second Gotch primer, which allows for exponential amplification of the Gotch fragments, while the ATAC fragments are only linearly amplified, as observed there in step 4. So fundamentally, Gotch was designed to surmount the limitations by using genomic DNA as a target of amplification instead of mRNA. So that allows genotyping of the loci irrespective of their genomic location, which is a big advantage, and can actually open the field to study mutations that are not necessarily located in coding sequences. So how did we assess the efficiency of this new method? Basically, we performed a cell mixing study using 2 different human cell lines that have distinct genotypes, in this case for the TP53 locus. So we perform a 1-to-1 mixing of the cell lines, and then we perform Gotch. After performing the experiment, we realized that using the ATAC data alone, we can identify both cell lines confidently, based both on canonical cell markers. For example, in the case of CA46, which is a lymphoblastic cell line, you can see increased accessibility of the early basal factor 1 gene, while in the health cell line, which is erythroblastic, you can see an increase of GATA1 accessibility. But in order to be very confident about the identity of the cell lines, we also used mitochondrial variants, which are mutually exclusive between the 2, just to orthogonally verify the identity of the cells. So our next step was to actually perform the Gotch genotyping calls. And what we observe is that when we project the genotype labels obtained by Gotch it results in very highly accurate. In this case, in this particular experiment, we had 99.7% accuracy, and we recover almost 50% of the cells genotypes. So this demonstrates that you can successfully genotype the cells at the same time as obtaining chromatin accessibility information. But we wanted to even further validate our genotyping call. So to try to obtain an orthogonal validation of this, we knew that the wild-type cell line contained an amplification at chromosome 9. So what we did was to perform copy number variation inference to show that the cells labeled as wild-type by Gotch actually carry this chromosome 9 amplification, while the mutant cell line showed no amplification at all. So these results kind of like gave us confidence that the Gotch approach was working effectively and with high accuracy. Now, I kind of already gave you the final result of the experiment, but to get to the genotype calls, we had to develop a set of analytical pipelines, which now are compiled together in form of an R package. Basically, what we do is to obtain the initial genotyping library, then perform a split of FASTQ and filtering, just for parallel processing to speed up the process. We identify those reads that contain the expected primer sequencing, and then we perform a whitelist matching just to keep those reads that have barcodes that are confidently mapped to the expected ones. Then after doing those 3 steps, we perform a read genotype assignment, and we provide an output as a summarized wild-type and mutant read counts per cell barcode. So this process can be performed in R and we parallelize using a SLARM cluster for increased efficiency. So when we did these first steps, what we noticed is that the genotyping reads had a bimodal distribution. And this bimodal distribution basically reflects cells that were effectively genotyped versus background noise in the data. So Sanjay Kutupalli, a very talented master student in the lab, developed an analytical pipeline to perform noise correction and genotype labeling based on these distributions. So after identifying thresholds for the noise and signal modes, we can calculate Z-score for wild-type and mutant reads and cluster the cells based on their read values. So this approach resulted in the high accuracy that we saw in the previous experiment, and it's very efficiently identifies genotypes from the Gotch libraries. Of note, we also developed an alternative approach using empty droplets to estimate background noise. And we saw that this method had a slightly decreased efficiency, although it was very consistent with the new method as well. So, but it had one small caveat that was it had increased difficulty in detecting heterozygous calls. So we stick to the first method based on the bimodal distributions for the rest of the paper. As an example, we just to address directly the heterozygous calls issues, we perform another cell mixing experiment using this time 3 different cell lines. One that is homozygous mutant, homozygous wild, other that is homozygous wild type, or a third cell line that is heterozygous for the JAK2 mutation. So when we apply Gotch to this 1-to-1-to-1 mixture, we still observe a high accuracy, 96.2%, and we genotype 63% of the cells. But most importantly, the set 2 cell line, which is the homozygous one, we found that we can correctly call heterozygosity in 35% of the cells. So Gotch can capture heterozygosity, but there was something else in this experiment that we noticed, that was that basically the efficiency of the percent of cells that we were capturing per cell line were very variable. But what we noticed is that the locus copy number, so that the copy number within the JAK2 site that we were targeting differed between cell lines as well. So when we basically tried to correlate these 2 features, we found that the locus copy number directly correlates with the fraction of cells genotype. And this makes sense because basically you have an increased amount of input material, and that increases your chances of actually capturing the locus during the PCR, the in-drop PCR step. But the most important part is that even if the locus copy number affected the efficiency of the capture, it did not affect the accuracy of it. So the Gotch remained highly accurate regardless of the copy number of the targeted site. So now that we verify the accuracy and the efficiency of Gotch, then we move on to applying Gotch to human samples to define epigenetic changes driven by somatic mutations. So in order to do this, we focus on myeloproliferative neoplasms, which we call MPN for short. MPNs are a group of hematological malignancies that are characterized by an overproduction of mature myeloid cells. They also present the risk of progression to what is called a spent phase that is characterized by marrow fibrosis and cytopenias. And they carry an increased risk of progression to acute leukemia as well. So all these MPNs have mutated constitutively active kinases that lead to independence of growth factors, just such as BCR, IBL fission, JAK2, MPL, or CALR mutations. So we next focus on primary myelofibrosis, which almost 50 to 60% of these cases are driven by JAK2 mutations. And together with essential thrombocythemia and polycythemia vera, these are the three main types of MPN that are driven by the JAK2 mutation. So we perform Gotcha in a set of 7 patients with myelofibrosis, and including 1 patient with longitudinal data that progressed from polycythemia vera to MF. So when applying Gotcha, we obtained 32,000 cells across different cell types. And based on the ATAC data, we basically cluster the cells and identify the cell types from the sample. It's important to notice that these samples were sorted for CD34 positive cells to enrich for hematopoietic progenitors. So here we show basically the integrated data set for all 7 patients with the progenitor subtypes identified using canonical cell type markers. So most importantly, when we project the genotyping data into the embedding, which was agnostic to the genotype, we managed to get genotype information for 60,000 cells, with just under 50% of cells genotyped for each sample on average. And we noticed that in the bar plot on the right, this is a major advantage of Gotcha, utilizing genomic DNA relative to other high throughput RNA-based approaches that were applying to studies, to previous studies before this. So here we can see that we have almost a 5-fold increase in the efficiency of genotype capture. So as the main driver of MPNs, it's well documented that the JAK2 V617F mutation results in a myeloid differentiation bias. So when comparing wild-type and mutant cells within the same samples across different progenitor clusters, we showed a clinically consistent lineage-specific fitness landscape. So we have an enrichment of mutant cells in the erythroid and megakaryocytic progenitors, which is the expected phenotype for these diseases. But we also analyze mutant cell fractions along, for example, erythroid pseudotime. So we can order the cells in pseudotime along a differentiation trajectory and ask how many mutant cells are within each of these quantiles. So in untreated samples, we found there is a steady increase in the fraction of mutant cells along erythroid commitment. And we also find similar things along the megakaryocytic pseudotime trajectory. But interesting, when patients are treated with JAK2 inhibitor, which is ruxolitinib, it results in a reduction of this bias and a more homogeneous distribution of mutant cells throughout the hematopoietic hierarchy. And of note, ruxolitinib treatment did not eliminate the mutant clones, which is in line with what is known about clinical responses. So Rux usually leads a symptom reduction in patients, but it doesn't reliably reduce the clonal burden or prevents disease progression over time. So next we focus on trying to see what to perform intra-cluster intrasample comparisons between wild-type and mutant cells. So for example, we used gene accessibility scores as a feature that we can interrogate with this method. And for example, we focus on the hematopoietic stem and progenitor cell one cluster, or HSPC1 here, shown here. And we see that there are a set of upregulated genes that belong to the inflammatory response pathway. And inflammation is one of the major hallmarks of this disease. So both the pro-inflammatory microenvironment as well as intrinsic pro-inflammatory keys can drive this disease. And here we're performing an intrasample comparison, and we're saying that that mutant progenitor cells have an increased inflammatory response compared to the wild-type counterparts that are within the same microenvironment. So there seems to be an intrinsic pro-inflammatory signaling of phenotype within these HSPCs. So another feature that we can interrogate with CICA-seq data is basically to indirectly estimate activity levels of transcription factors by measuring the accessibility of the DNA binding motifs for each factor. So when performing this differential motif analysis in the HSPC1 cluster, we see that transcription factors involved in NF-kappa B and SHUNFOS pro-inflammatory signaling are increased in the mutant progenitors, which is consistent with the enrichment that we saw in the inflammatory response pathway as well. And in addition, these motifs correlate with STAT1, 3, and 5 accessibility, which is shown on the panel on the right. And these are the canonical downstream targets of the constitutively active JAK2 V617F mutation. So this could be a link from the mutation towards the pro-inflammatory phenotype in an intrinsic manner. Since Gotch it allows for intra-patient comparisons of wild-type and mutant cell, these findings are actually revealing a cell-intrinsic pro-inflammatory epigenetic landscape that is already present at the earliest point of hematopoiesis. Then by leveraging a longitudinal sampling from a patient that had a polycythemia vera that progressed to MF, we show that Jun and NF-kappa B motif increase is already observed in mutant cells in the polycythemia vera stage, and it remains higher in the MF. So even before progressing to myelofibrosis, there is already an intrinsic pro-inflammatory phenotype that is observed in the polycythemia vera stage. So, and another capability of Gotch is that we can perform these comparisons within each cell type. So next we explore HSPC2, which is a slightly more differentiated stage of the stem cell compartment. And we found that differential motif accessibility reveal a much stronger pro-myeloid signature comprised of TFs that include, for example, LMO2, TAL1, and RUNX1, which are increased in the mutant to a higher extent than what we observed in the HSPC1 cluster. So this further supports an epigenetic priming towards myeloid differentiation that begins in early hematopoiesis. In addition, we observed a few transcription factors that function in TGF-beta signaling. So not only reinforcing the intrinsic pro-inflammatory signature that we saw before in the HPC1 cluster, but also highlighting that it's a cell type specific phenotype that can be identified through Gotch. As an orthogonal validation, we also leverage a very elegant mouse model developed by Andy Dunbar and Robert Bowman from the Levin lab that allows for a sequential knock-in and knockout of JAK2 V617F. As these demonstrate that the pro-inflammatory phenotype is dependent on the constitutively active form of JAK2. So next we looked into the other myeloid lineages because we wanted to know whether these cell type specific phenotypes were also different in other progenitor subtypes. And when assessing differential motif accessibility of early erythroid progenitors, the mutant cells exhibit an enrichment in STAT5A and 5B, which is to some extent expected because they are downstream of JAK2 targets. But we also observed a decrease in the accessibility of BCL11A, which is highlighted there in red. And this particular transcription factor is known for its inhibitory role of the gamma globin, which is a component of fetal hemoglobin. And there were some observations that PV and MF can actually, patients can actually show increased fetal hemoglobin levels. So we were intrigued by this observation. So another feature that we can interrogate with single-cell ATAC is the co-accessibility of different regulatory regions. So here we looked into the HBG1 enhancer site, and we show that it actually shows decreased co-accessibility in the, increased co-accessibility in the mutant cells, thus probably indicating an increase of the HBG1 gene itself. So in order to see whether this increased co-accessibility, and of note, a different peak within the locus control region did not show any differences between wild type and mutant cells. So this seems to be a region-specific effect. So next we looked into the HBG1 gene itself, and what we observed is that we had an increase in the accessibility of HBG1 within the mutant cells, together with an increase in the fraction of cells that show this accessibility and the overall accessibility itself. So this implies that there is already an epigenetic priming towards increased fetal hemoglobin production driven by the JAK2 mutation. Again, by leveraging the longitudinal sample, we show that both the decrease in BCL11A and the increase in the HBG1 gene accessibility is already observed in PV way before this, there is a progression towards myelofibrosis. So finally, we wanted to look into the megakaryocyte progenitors, MKPs, because these are linked to the profibrotic environment in myelofibrosis. So we performed differential motif accessibility analysis, and that reveal, again, a cell intrinsic pro-inflammatory phenotype that is restricted to Jun and Foss motifs, in contrast with the more broader phenotype that we saw earlier in HSPCs. And we saw this through increased footprinting as well. So in addition to this, we show increase of TGF-1, which is a member of the TGF-beta signaling, which has been associated with the profibrotic activity of MKPs. So after describing the system within patients, we next ask whether can we expand Gotcha to include multimodality single-cell sequencing. So can we increase the layers of information that we capture during these experiments? So we aim to expand Gotcha's capabilities using ASAP-seq or ATAC with select antigen profiling by sequencing, which was developed by our collaborators here at the New York Genome Center. And this allows, basically it's a high throughput multi-omic method that allows through fixation of whole cells prior to the 10x single-cell ATAC. Sorry. It enables to measure jointly chromatin accessibility and protein expression, and it has the additional advantage that increases the capture of the mitochondrial genome. So we apply Gotcha ASAP to one sample, or two samples actually, but I'm showing only one sample for illustrative purposes. So this demonstrates the Gotcha genotyping method is fully compatible with ASAP, with the ASAP protocol. Although we managed to measure protein levels at cell surface protein levels in particular, we did notice that there was a slight drop in the fraction of genotype cells, which I think is possibly due to the fixation conditions that are required for ASAP-seq. However, we also noticed that we could leverage mitochondrial variants that were occurring in phase with the JAK2 B617F mutation, missing genotypes. So for that process, Tamara Prieto, who is a very talented postdoc in random forest classifier that allows imputation of missing genotypes. So when we perform the genotype imputation, given the higher coverage of the mitochondrial genome, it allows for a dramatic increase in successfully genotyped cells in the sample. So while we originally obtained around 20% genotyping with Gotch ASAP, the imputation using phased mitochondrial variants increased the effective genotyping rate of the sample to just over 90% of all cells. In addition, we were able to measure cell surface protein expression. So we perform a differential analysis and we reveal an enrichment in CD90 in a subcluster of HSPCs that is dominated mostly by mutant cells. So as you can see here, a pseudo fact sorting plot showing CD34 and CD90 expression shows that CD90 high, CD34 high cells are mostly composed by mutant progenitors. So 93.1% of the cells are called mutant by Gotch. And since with Gotch we also have chromatin accessibility, we can link this increased protein expression indirectly to the increased accessibility of the gene itself and of a proximal peak upstream of the gene itself. So in conclusion, we show that Gotch provides a high throughput genotype to epigenome linkage at single cell resolution. We show that it surmounts some of the limitation and dependencies from RNA-based methods that are based on gene expression and location. We show that it's compatible with intact cells or isolated nuclei as input, and they're amenable to multi-omic integration. It can synergize with mitochondrial variant calling to further improve genotyping. And we think it's compatible, we know it's compatible with single cell assays that depend on in-droplet PCR, such as Genotype and DT-seq, etc. And then importantly, our investigation on JAK2 B617F in human fibrosis showed that there are cell intrinsic and cell type specific shifts in epigenetic landscapes. So we show that HSPCs have a pro-inflammatory and myeloid bias signatures. We show that the erythroid progenitors exhibit increased STAT and erythroid motif accessibility, as well as aberrant regulation of the gamma globin locus. And we show that MKPs demonstrate a robust JUNFO signature, which supports their role in myelofibrosis. So with that, I'd like to first thank my mentor, Dan Landau, and especially Robert Myers, who is a very talented PhD student in the lab who led this work with me and is actually presenting his thesis defense tomorrow, as well as our funding sources, including the American Society of Hematology and all our collaborators. So lastly, I want to thank you for listening, and I'll be happy to take any questions.
Wonderful. Thank you so much, Franco, for sharing your work with us. And we'll move on to our Q&A. I want to start with a question about what is the feasibility of using Gotch on archival frozen tissues or tumors? Is that something in the works, or?
That is a great question. And that's one of the big advantages of using the nuclei-based input, like what we call the standard Gotch. So not for ASAP, because ASAP requires whole cells and fixation, but if you have frozen archives that contain, like you can isolate nuclei from, there is no reason why Gotch will not work on those that I can think of. It may require some troubleshooting as always, but it should be feasible.
Okay, feasible, but you haven't tried it yet. Well, we'll see.
So,
to
be clear, yes, some of the samples that we have in our paper were retrieved from frozen archives actually that we performed standard Gotch. So yes, it's feasible.
Another question that has come in is, have you done any comparison of your MF patients with JAK2 mutations to patients with other mutations like MPL or CALR?
That's a great question. And the short answer is we haven't yet. We hope in the future to, of course, and we hope people to broadly adopt this technology and to explore different mutations. And that also brings up a question of we noticed that we targeted different, for example, in our cell mixing experiments, we target different loci, and we saw a similar efficiency on the capture. So I would expect that if as long as the region surrounding the mutation is not extremely complex for designing the primers to capture the mutation, it should be feasible to explore those as well using this method. Absolutely.
Great. One last question here. Have you compared patients who start off with other diseases such as polycythemia vera and progressed MF with patients who have primary MF?
That's also a great question. So we only have one longitudinal sample so far. We're struggling, not struggling, just trying to get more because yeah, it's not an easy task, but we haven't directly made that comparison, and that's a very interesting comparison because we observe, for example, that in PV you can already see some of the features that you see later in MF. The question is, does primary MF also have these same features or not? And those are the kind of questions that you can address with the technology as well. So it will be very interesting for people to, to think about these questions and in the context of how to apply the technology. And just want to make a little bit of marketing. Like we want this to be easily applicable and to be adopted for as many people as possible. So we have our analytical pipeline on GitHub now is in beta testing. So feel free to try it out. And then the primer design is the detail in the preprint as well. So you're more than welcome to give it a try as well.
Well, wonderful. Thank you for offering that to the community. And yes, thank you again for your talk. It's been very exciting. The seminar has been recorded and will be available soon on YouTube and on our website, multimodalintegration.org. You can also follow us on Twitter at segs_ICA or check our website for the latest announcements regarding upcoming talks and events. And with that, I'd like to thank everyone for joining. Thank you so much again. And we hope to see you at our next seminar.